Hello everyone, my name is Chase, and today I will be presenting a study on chronic exposure to synthetic food colorant Allura Red, also known as Red 40. Now, I'm sure you all have heard of Red 40. There's tons of media surrounding the topic, with these types of dyes being in pretty much every food and can potentially be harming us. So this study will look into the effects of this particular food dye on the microbiome. Now, the authors covered a wide range of various tests and experiments that all sought to find a connection to Allura Red exposure and potentially exacerbated colitis development. I'll talk about the specifics on the next slide with some background information, but in essence, they were trying to see if Allura Red causes gut inflammation by increasing serotonin levels. Now, this increase in serotonin would be a telltale sign of IBD, or inflammatory bowel disease, which is what colitis is. And with mice being the test subjects, we could apply the findings to the human microbiome and see if Allura Red actually is harmful. Alright, so some background information, starting with Allura Red. As I said before, it's Red 40, which, if you don't know what Red 40 is, I recommend reading your nutrition labels. It's a FDA-approved petroleum-derived azo, which just basically means it's a highly processed synthetic dye. We have it in pretty much everything nowadays. It's in Fruity Pebbles and Fruit Loops. It's in Skittles and Jello, pretty much all candy. It's in Gatorade, any fruit punch flavored drink. There's even a study done in 2007 that found Allura Red in red meats like salmon and beef, and it was used to enhance the color and maintain a fresh appearance for the meat, which is crazy. So, yeah. Next we have serotonin, and this is a neurotransmitter that gets signaled during times of inflammation or epithelial barrier function. They're specifically in the gut, about 90% of it. So it's a perfect way to tell if there's been some damage in the gut microbiome if these are abundant. Now in the study, they have a whole range of denominations for this compound. Its actual name is 5-hydroxytryptamine, or 5-HT, and they often talk about the specific pathway of its activation. So there are various enzymes that are directly related to its synthesis and inflammation as a whole, and thus also need to be accounted for. To list two, there's tryptophan hydroxylase 1, or TPH1, which is the enzyme that actually produces serotonin. And there's also enterochromaffin cells, or EC cells. These are the cells in the gut that release the serotonin when it's been signaled for. There's a few more, but they're very specific to the actual study, so I'll get into them when we get into methodology. And lastly, we have colitis, which, as I mentioned, is a form of inflammatory bowel disease. It's a generalized term for irritation and damage to the colon or large intestinal lining. In humans, this can cause stomach pain, diarrhea, and weight loss and fatigue. Diet has been proven to be a significant factor in the causation of IBD, so it's important to look into the foods that we eat and try to minimize the potential harm that we could be unknowingly doing to our gut health. Okay, so now onto the actual study. The researchers used a mouse model to simulate the Allura Red and colitis relationship. They separated mice into two groups, a control group and a custom group. Both groups were fed a TechLad irradiated soy protein rodent diet, which is just a big term for your typical hard pet food, just for rodents. However, the custom group had their diet coated in Allura Red as well as in their drinking water. The amount of Allura Red distributed amongst the custom diet was akin to the amount that is acceptable in humans, which was about 7 milligrams per pound of body weight. And the mice were fed this diet and monitored for 12 weeks. Now, the researchers induced colitis into all of the mice using dextrans sodium sulfate, or DSS, for 7 days in their drinking water. What DSS actually does is it elicits a strong inflammatory response by disrupting the intestinal epithelial layer. This creates these little perforations in the intestinal lining that allow surrounding mucosa and luminal bacteria to get inside the lining and start causing tissue damage. This then releases cytokines and elicits other responses that the researchers assessed when examining the severity of this disease. Furthermore, we're going to get more into the specifics of the various monitoring techniques that were used to evaluate the severity of inflammation to best compare the two groups. Okay, so first we have the Disease Activity Index, or DAI. This was a scoring system that combined the individual scores of weight loss, stool consistency, and fecal blood. So points ranged from 0 to 4, 0 meaning there was no weight loss, stool was normal and had no blood in it. 2 meant there was about 5 to 10% weight loss, stool was loose and had trace amounts of blood. And 4 meant more than 20% of weight loss, stool was considered diarrhea, and there was detectable blood around the anus. This system was used for the 7 days of DSS induction. There's also a macroscopic score that we'll see on the results slide. This was given based on the exact level of colonic bleeding from the DAI test. Okay, on to histology. So the colons were washed with PBS, which is a saline solution that matches bodily pH. It keeps everything nice and neutral. And then it was cleaned with ethanol and then stained with H&E. H&E is just a cell-specific dye that helps visualize everything. They looked at the colon architecture and scored it from 0 to 3 in terms of crypt distortion or complete loss of crypts. Crypts being these little pits within the intestinal epithelial layer. They're used mainly for secretion of stem cells during inflammation. They also looked at the degree of inflammation, cell infiltration, and scored that from 0 to 3. The scores reflected either no immune cells within the stained tissue, some immune cells present, or severe infiltration. So there was like a whole bunch of immune cells in it. The amount indicating the level of inflammation within the tissue. They also scored muscle thickening of the tissue from 0 to 3, using the base of the crypt's location to assess the amount of thickening. And lastly, they scored goblet cells from either being present or absent. These goblet cells are found in the intestinal lining and are used to secrete mucus, which I think we all know what mucus is. It's just a protective lubricating layer that coats the tract and traps bacteria. Seeing if the goblet cells have been obstructed or not helps determine the severity of colitis inflammation. Okay. Now for these next few parameters that were evaluated, they are all the consequences of generalized inflammation. What I mean by that is that higher levels of each one of these factors are all an indication of inflammation in the gut microbiome. So to start listing them, we have MPO, otherwise known as myeloperoxidase. This is an enzyme that is found in neutrophils. When these neutrophils are activated to respond to inflammation, they release this enzyme that combats the bacteria that is embedded within the tissue. The H&E dyed colon allows for the visualization of these neutrophils. Next, there's the presence of PMLC, which is phosphorylated myosin light chain, which is an indicator of the activation of myosin light chain kinase. This protein is usually seen within the intestinal lining when the gut's permeability has become too loose and too much stuff is crowding the lining and causing damage. Thus, PMLC will come in to contract the junctions and seal it so the permeability is not so permeable. This correlates to DSS when I mentioned how the induced colitis will cause perforations or holes in the lining, making the gut become toxic. So they were able to view these proteins using Western blotting. For this next one, in my opinion, this is probably the most important graph that we'll see in the results slide. It pertains to this next test, which was used which used ELISA software to test for serotonin levels. As I spoke about in the background slide, this is pretty much the key thing to look for when checking inflammation severity, and this test just straight up looks for it, using the colon tissue samples and gives an approximate value. And the final inflammation checker that also used ELISA in the presence of, or is the presence of cytokines. Cytokines are the health communicators in our body that come in to signal T cells, such as the neutrophils I talked about earlier, to regulate inflammation. And they tested for 3 types of cytokines, those being IL-6, IL-1 beta, and TNF alpha, all of which are indicators of something going on in the gut microbiome. Now for this last method, the researchers used PCR to look for the expression of mRNA. mRNA is important because it tells us about gene activity, what genes are being expressed, which tells us something about the inflammatory response. The main gene being TPH1. This is the gene I mentioned in the background slide that creates the hydroxytryptophan, or serotonin. Once again, a telltale sign of possible inflammation. Okay, now with all of our methodology addressed, we can start looking into what the results found. So for the results, there are a plethora of charts and diagrams that can be a lot to look at. I'm going to just pinpoint the important monitoring factors that I highlighted prior, so that there's no confusion and we can just have a more direct explanation for things. Okay, so in retrospect, the results point to the notion that colitis development is increased by Allura Red. Why is that? So starting here with this table 1C, you can see that there was an upward trend of the disease activity index in 4 groups. The 4 groups being the chow diet or control group, the Allura Red diet group, the colitis induced group, and the colitis/Allura Red exposed group. The colitis/Allura Red exposed group is shown to have a higher DAI than any other group, as well as the Allura Red group having a higher DAI than the control group. This goes to show that the combined scores of weight loss, stool consistency, and fecal blood was higher in the mice that were given Allura Red combined with colitis. In other words, the dye caused weight loss, rectal bleeding, and loose stool. And we actually can see that in the individual scores. For example, with the weight loss score, here in figure 1b, we see that DSSS plus AR, or colitis and Allura Red group, is the lowest weight of all the groups. As well as here in table 1D, the macroscopic score, which showed bleeding levels being higher in the DSSS plus AR group. Okay, the next thing we're going to look at is the colon scoring, or histological scores. Here in table 1J, we see once again the DSSS plus AR group has a significantly higher score than the other groups, as well as the AR only group being higher than the control group. This goes to show that the overall structure of the colon was degraded in terms of goblet cell degradation, colon shortening, and crypt loss. And then here in 1E, they actually show the picture of the colons to support this colon shortening terminology. And you can literally see how it gets less and less with the addition of Allura Red, and then even more with colitis exposure. And then the shortest colon is the one here that has Allura Red exposure as well as colitis exposure. And then right here in table 1F, they have the actual numbers in centimeters, so you can visualize everything better. Okay, now I want to turn your attention to table 1I. This is another chart I would like to highlight. This is the H&E staining technique that researchers used to visualize the actual degradation of the intestinal epithelial layer. In the control group, right here, top left, it looks like a normal tissue. Then there's Allura Red, which this white portion here seems to enlarge. And then here in the colitis model, it looks somewhat similar to the control group. I couldn't actually determine a difference. nor do the researchers actually reference this portion. However, the Allura Red with colitis exposure does show a major difference, with this entire outer layer here now intruding into the epithelial layer. This is the effects of gut inflammation, where the crypts I spoke about before will degrade and create holes in the lining, which ruins the permeability of the gut. You then have these inflammation cells like the neutrophils with MPO coming in and intruding into this layer, which messes things up. And that actually lines up perfectly with the table right here, which is 1K. We can see a higher amount of MPO within the tissue in the mice exposed to Allura Red and colitis. And lastly, we have these three charts down here on cytokine production: IL-6, IL-1 beta, and TNF alpha. Each one of these shows much higher expression in the DSS plus AR group, which is shown to prove that it has worsened colitis. All in all, these charts drive home the point that the DSS plus AR group of mice had the most inflammation response factors by a long shot, further proving that colitis development was increased. The next fact that was shown from the results: gut inflammation is elevated with the addition of Allura Red. Now, all these charts pretty much show the exact same findings from the previous slide. However, the researchers have isolated the mice groups in these charts to just two: the control diet group and the Allura Red diet group. This was done to focus on how the ingestion of Allura Red alone, without induced colitis, will still cause gut inflammation. Macroscopic score is higher with Allura Red, the histological score is higher with Allura Red, the MPO production is higher with Allura Red, and all cytokines are expressed more. Once again, we see the exact same trends as before, with an increase in inflammatory responses now due to the colorant alone. Now, for 1G here, I didn't really talk about the methods that they used here because the outcomes of it was the same as the ELISA tests, which I found to be more pertinent to discuss. But I'll just quickly highlight what's going on here. So, this is immunofluorescence staining. It's a method of visualizing 5-HT levels using Alexa Fluor 568. This is just a bright red-orange dye with a fancy name. And if you look here, you can actually see the 5-HT cells in the colonic tissue. I personally think the ELISA software showcased it better with the actual numbers because I can't really tell a difference between the control diet and the Allura Red diet. It seems to be roughly the same amount of red dots here. But as we're going to see in the actual ELISA test, the serotonin levels were much higher in the Allura Red group of mice. This here is just a continued slide to further prove the point of gut inflammation becomes elevated with Allura Red. I wanted to add this chart here, which is chart 4A, because it has some interesting findings. This chart shows the PCR tests that looked for mRNA expression, which, as we know, causes gene activation of TPH1, as well as activation of MLCK. When the tissue was pre-treated with TNF alpha, Allura Red exposure significantly boosted MLCK levels. When TNF alpha was not involved, Allura Red didn't have much of an effect. What does this mean? This goes to show that during inflammation, when TNF alpha and other cytokines are being expressed to help regulate the damages, Allura Red's presence activates the MLCK pathway, which suggests that Allura Red causes damage to the epithelial layer and ruins permeability. Because the only reason MLCK would be activated is to tighten those junctions within the layer using PMLC. Because if the junctions are wide open, mucus and bacteria is getting in, and that's why the cytokines are being expressed. So this is just more evidence on how Allura Red is directly related to elevated gut inflammation. Okay, and the last point I'm going to highlight is the importance of serotonin and its relationship with Allura Red. So this was the chart that I said was probably the most important for proving the Allura Red, that proving Allura Red causes gut inflammation. Because these are the readings that ELISA provided, which show the actual serotonin levels of 5-HT, as they are labeled right here. Almost all these enzymes and compounds I've talked about here have some kind of relationship with either the activation and production of this neurotransmitter or are released because of its presence. And as we see here, the Allura Red diet group of mice demonstrate higher levels of 5-HT compared to the control diet. Clear evidence that serotonin, the primary gut inflammatory regulator, is directly linked to Allura Red exposure. The colorant comes in and causes TPH1 to rise. TPH1 then goes on to produce serotonin. The EC cells release this serotonin. The serotonin then activates the inflammatory cytokines TNF alpha and IL-6 and IL-1B or beta, and then they go off and activate a whole bunch of inflammatory enzymes and proteins to help try and regulate the inflammation. So this graphic kind of sums up everything that the researchers have determined. So you have your normal intestinal epithelial barrier here with functioning goblet cells, functioning EC cells, and you have a normal serotonin level, everything's good. Then you add Allura Red to it, and there's an immediate increase in serotonin production. And thus, with that increase, comes an increase in inflammatory cytokines. Things are starting to flare up. And over here, if you have colitis and Allura Red exposure, the epithelial barrier's integrity is compromised, causing a disruption of the gut microbiome. Alright, so to conclude, the results showed that chronic exposure to synthetic dye increases susceptibility to colitis. We see this in all the graphs that showed DSS plus AR values, and those values were higher than any other group, be it DAI, colon scoring, and so forth. The Allura Red increases serotonin synthesis, promoting inflammation. The other point, Allura Red causes gut inflammation. We see this in the control versus Allura Red diet graphs that demonstrate higher cytokine release, MPO, histological scores, as well as mRNA expression, all of which proving some form of epithelial barrier damage or inflammation of the gut. And lastly, serotonin has a direct relationship with Allura Red, meaning that when Allura Red was added, serotonin levels rose. The absence of Allura Red signifying normal levels of serotonin. All in all, Allura Red can exacerbate inflammation of the gut and even cause a degree of inflammation on its own without colitis. Alright, now with all of these conclusions determined, we can begin looking into further studies and more discussions regarding food dyes and inflammation. The first question that I have is, do all food dyes increase serotonin production? Would we see the same symptoms that were displayed here from ingesting other dyes, such as tartrazine, which is yellow 5, or blue 1, both of which are present in a whole bunch of food and drinks as well? So are they causing harm to our gut microbiome the same exact way that Allura Red was shown to? The next question I have, does Allura Red or other food colorants affect other neurotransmitters like dopamine or epinephrine? Granted, serotonin is affected primarily because 90% of it is produced in the gut. However, dopamine and epinephrine are also present there. Maybe not as much, but are they also affected the same way serotonin is? Another question I had, what are the long-term effects of Allura Red? Because the study only fed them this diet and studied them over a course of less than a month. And if we look at a more long-term time span, would these effects still persist, and to what degree? Another question, are people with gut sensitivities more prone to complications with food colorants? People with IBD, people with allergies. What is happening in their intestinal lining when exposed to Allura Red or other food dyes? And the last question I have, with everything being said here, is this enough to discontinue the usage of food colorants? Does this convince you to stop drinking Gatorade or eating candy that is filled with dyes? A lot of countries in Europe have already banned a lot of these dyes in food, so are we next? And here are my references. Thank you so much for listening, and I'm looking forward to reading the discussions in the JClub.